My dear students, in this video, we are going to see the contributions of three important scientists, Beijerinck, Winogradsky, and Metchnikoff. I know it is a little difficult to remember and pronounce these names, but after knowing their contributions towards microbiology, it wouldn't be difficult to remember these names. In fact, when a whole group of scientists were more interested in medical microbiology, the microorganisms which are responsible for human diseases, these scientists dared to move away from that known field and given very important contributions towards microbial ecology, environmental microbiology, immunology, and also virology. Let us see their work one by one. The scientist you're seeing here is Martinus W. Beijerinck. He was a Dutch microbiologist and a botanist. He was one of the founders of virology and environmental microbiology. He had written over 140 papers on different topics in microbiology, botany, chemistry, and also in genetics. Perhaps the most important contribution of Beijerinck is the discovery of a new infectious agent called a virus. In fact, before him, there were two scientists who were working on this particular infectious agent. First one was Adolf Meyer. Adolf Meyer was working on a disease called tobacco mosaic disease. In his time, tobacco processing industry was a very much profit-making industry, and he was studying on a particular disease that was affecting this tobacco plant, that is tobacco mosaic disease. He tried to isolate the infectious agent which was causing this disease. He used to take the infected sap and he used to inject it in a healthy tobacco plant, and observed that the same type of symptoms was seen in the healthy plant, just like it was there in the infected plant. And he assumed that this infectious agent is basically a bacterium. Though he assumed it, he could not grow it in the laboratory. He could not prove that a specific bacterium is responsible for this disease. And the next person who was doing the same studies on tobacco mosaic disease was Ivanovsky. Ivanovsky was doing a lot of experiments in his laboratory to prove the infectious agent of this tobacco mosaic disease and to isolate the organism. So just like Adolf Meyer, he took the sap of this infected plant or infected leaf, and before injecting it into healthy leaf, he filtered it first. So basically what he thought was, when you filter this sap, whatever bacterium that is responsible for the disease will get filtered out, and you will get a sap which is free of the infectious agent. So after filtering this sap, he again injected it in healthy leaf, and to his utter surprise, he found out that even after filtration, the sap could cause the same type of infection in a healthy leaf. So basically, what he had to assume was the infectious agent was a filterable substance. Next, what he did was he took this infected sap and tried to isolate the organism in the laboratory by growing it in a nutrient agar plate. Again, he observed that he could not grow the organism present in the sap in his laboratory plate. So with these two findings, he found out that the organism that is responsible for tobacco mosaic disease is basically is basically a new infectious agent which is a little different from the bacterium. And it is finally Beijerinck who recognized the causal agent of the tobacco mosaic disease as a completely new type of infectious agent. In fact, he said that the infectious agent is a filterable organism which is present in the liquid form, and both Ivanovsky and Beijerinck have been credited with the discovery of this new infectious agent, which we now call virus. They didn't call it as virus, but initially it was called with some other name, and now we know that it is the virus which is responsible for tobacco mosaic disease. So the first discovery of a viral agent which causes a plant disease was done by Winogradsky and Beijerinck. So whatever I said it is there in this YouTube link. Please go through this YouTube link to see the animated version of what I said now. So on the basis of his laboratory observations, Beijerinck called this organism which caused the tobacco mosaic disease as Contagium vivum fluidum, which literally means contagious virus fluid, a term coined to convey his concept of living infectious agent in fluid form, that is in a non-cellular form. This was a revolutionary idea at a time when always life is connected with cellularity, that is only a cell is called a life. So he said this is a living infectious agent which is non-cellular in nature, which is in fluid form. Let us see his other contributions. One of the greatest accomplishment of Beijerinck is the development of enrichment culture technique. In fact, he did it simultaneously with Winogradsky. And what is this enrichment culture technique? See, when you want to isolate microorganisms that occurs in nature, for example, in soil or in water, most of the times these microorganisms will be present in very small numbers. So it is really difficult to isolate them in pure form, that is in pure culture form, in the laboratory and study them. So what he did was, he developed certain techniques like adding some special nutrients or creating some special environment in the laboratory so that these organisms which are present in nature will grow specifically and will form colonies so that you can get them in pure culture and study them. For example, if you want to isolate specifically cellulolytic bacteria, what you have to do is you add cellulose as the carbon source in the medium, in the nutrient agar medium, so that the cellulolytic microorganisms will grow more in numbers and you can see those colonies more in numbers in the laboratory plate. So this is what we call enrichment culture techniques. So using this enrichment culture technique, Beijerinck was able to isolate a lot of special microorganisms like sulfate reducing bacteria, urea bacteria, oligonitrophilous microorganisms, denitrifying bacteria, lactic and acetic acid bacteria. In fact, using this enrichment culture technique, he isolated organisms like Rhizobium leguminosarum, which is a bacillus that fixes atmospheric nitrogen and causes the formation of nodules on the roots of leguminous plants. You might have even studied this in PUC, called symbiotic microorganisms like Rhizobium. They form nodule with higher plants like leguminous plants, and inside this nodule they do a process called nitrogen fixation, and by means of this they give something to the plant, that is a nitrogen source, and they also get something out of the plant, that is usually a carbon source. And another nitrogen fixing bacteria was characterized by him. This is a new group of nitrogen fixing bacteria that he had isolated and studied, that is Azotobacter. Unlike Rhizobium, this is a free living nitrogen fixing bacteria which is present in the soil. And he also named a new genus called Aerobacter, of which he distinguished 4 different species. That is, under one genus he could observe and differentiate 4 different species. That is the new genus Aerobacter. Beijerinck also discovered a new phenomenon called anaerobic respiration, which is carried out by the bacterium. What is this anaerobic respiration? See, we know most of the life forms on earth carry out aerobic respiration, including human beings, where we take in oxygen. Where does this oxygen go? The oxygen goes to the cell where it acts as the terminal electron acceptor in the electron transport chain. Right? So this is where oxygen is going to be essential in aerobic respiration. But what if there is an atmosphere where there is no oxygen? Doesn't the cell carry out respiration in such an environment? Yes, there are certain cells, there are certain microorganisms which grow in an anaerobic environment or oxygen-free environment. So if oxygen is not there, they are using some other source as the electron acceptor. And that could be anything like a sulfate or a nitrate. So if sulfate is used as the terminal electron acceptor, we call that respiration as sulfate respiration, or you can also call it as sulfate reduction, a form of anaerobic respiration. So this discovery was made by Beijerinck that sometimes in the absence of oxygen, sulfates also can act as terminal electron acceptor, resulting in anaerobic respiration. And this discovery had an important effect on our current understanding of biogeochemical cycle. So we know in nature, minerals keep on getting recycled in different forms. Sometimes it will be in the reduced form, sometimes it will be in the oxidized form. For example, if you take nitrogen, sometimes it will be present as proteins, as amino acids, that is in the reduced form, whereas sometimes it will be present in the oxidized form, in the form of nitrates or nitrites. So this is what we call biogeochemical cycle, which is a very essential part of the nature of the environment. So this biogeochemical cycle, if it is happening properly in nature, it is mainly because of the different microorganisms which convert one form of substance to another form, which converts a reduced form into an oxidized form and an oxidized form into a reduced form. So this discovery again was made by Beijerinck, and he also isolated and described another bacterium, sulfate reducing bacterium called Spirillum desulfuricans, which we now call as desulfovibrio desulfuricans. So his contribution is very much recognized here in the process of anaerobic respiration. Apart from his contribution towards environmental microbiology, he also investigated the luminous causing bacteria. He obtained the first pure culture of green algae, that is Chlorella, and he also studied the lichens. And his studies has been noted on several yeasts and also the butyric acid and butyl alcohol bacteria. The person you are seeing here is Sergei N. Winogradsky. He was a Russian microbiologist. His contribution was mainly towards environmental microbiology. In fact, it could be explained under three topics: his work on nitrogen cycle, his work on a new process called chemolithotrophy, his most noted discovery in fact, and the Winogradsky column. Before that, we must first understand a little about nitrogen cycle. You will be studying this in detail in your later semesters. But for now, let me just brief you about nitrogen cycle. See, what happens is the atmosphere contains 70% of nitrogen gas. And this nitrogen will first get fixed in the soil by means of a process called nitrogen fixation, and gets converted into ammonia or ammonium. This is carried out by a group of bacteria called nitrogen fixing bacteria. And once ammonia is formed, this ammonia will further be oxidized into nitrites and also nitrates. This is carried out by nitrifying bacteria, which is present in the soil by means of a process called nitrification. And then once nitrate is accumulated in the soil, this nitrate will be further converted to nitrogen and gets released in the atmosphere in the form of nitrogen gas. This process is called denitrification, which is carried out by another group of bacteria called denitrifying bacteria. So this is a classical example for biogeochemical cycle, where how the nitrogen gets converted into different forms in the environment. The atmospheric nitrogen gets reduced to ammonia and ammonium. and this ammonia gets oxidized into nitrates, and these nitrates further gets reduced into atmospheric nitrogen. And you can see for this whole process many different groups of bacteria are responsible. Here we have nitrogen fixing bacteria, here we have nitrifying and nitrosifying bacteria, here we have denitrifying bacteria. So all these microorganisms which are present in the soil are responsible for biogeochemical cycle, especially the nitrogen cycle that we are talking here. So Winogradsky was in the University of Zurich when he was doing studies on nitrogen cycle, and there he discovered two new microorganisms which are carrying out nitrification process. As I said, nitrification is a process where the ammonia gets oxidized into nitrite and then to nitrate, and he discovered two new genera which were doing this nitrification. One was Nitrosomonas and the other one Nitrosococcus. And apart from that, he also identified an obligate anaerobic bacteria, that is Clostridium pasturionum. This is a free living bacteria which is capable of fixing atmospheric nitrogen. See, unlike Rhizobium, which fixes atmospheric nitrogen in a symbiotic relationship inside the root nodule, this organism, Clostridium pasturionum, this lives freely in the soil and in an anaerobic environment and fixes atmospheric nitrogen. So this bacteria was isolated and studied by Winogradsky, and Winogradsky is also responsible for understanding the role of microorganisms in other biogeochemical cycles such as phosphorus and sulfur cycles. Winogradsky also discovered a new process that is happening in the nature, that is chemolithotrophy. What is chemolithotrophy? To understand this, we must know what is photoautotrophy. We know photoautotrophy is a process where the plants or the photosynthetic microorganisms use light as the energy source, and they use carbon dioxide as the carbon source, and they carry out photosynthesis process, right? So this is what we call photoautotrophic type of nutrition. Whereas we also have another type of nutrition called chemolithotrophy. What is the difference between this and this? See, in chemolithotrophy, we do not use light as the energy source. Instead, we use some inorganic compounds as the energy source. In fact, here, the inorganic compound that is used as energy source could be reduced compounds like hydrogen sulfide. And just like photoautotrophy, here also carbon dioxide is used as the carbon source. So this is also an autotrophic type of nutrition, but the only difference is instead of light, we use some reduced inorganic compounds as energy source. And this type of chemoautotrophy, or you can also call it as chemolithotrophy, is carried out by a special group of microorganisms called chemolithotrophs. And these chemolithotrophs were first discovered by the work of Winogradsky. So he discovered the first known form of lithotrophy during his research with an organism called Begiatova in 1887. And he also reported that Begiatova oxidized hydrogen sulfide as an energy source and formed intracellular sulfur droplets. So this is another contribution of Winogradsky I would like to mention, that is a Winogradsky column. So ಇದು ಒಂದು glass jar ಇದು. ಈ glass jar ಅಲ್ಲಿ he created different gradients by including different types of nutrients in different layers. He could grow different variety or different groups of microorganisms in the same place, and he could explain the metabolic character of all these microorganisms. For example, ಇಲ್ಲಿ ನೋಡಿ ನಿಮಗೆ ಲಾಸ್ಟ್ ಅಲ್ಲಿ cellulolytic microorganisms grow ಆಗುತ್ತೆ, and anaerobic microorganisms will grow here. Here you will find green photosynthetic microorganisms growing, purple photosynthetic bacteria growing, and top layer you will see cyanobacteria and algae growing. ಇದೆಲ್ಲ different groups of ಏನೇನ್ types ಇದೆಯೋ environment ಅಲ್ಲಿ ಆಲ್ಮೋಸ್ಟ್ ಎಲ್ಲ ಗ್ರೂಪ್ಸ್ ಅನ್ನು ಇನ್ಕ್ಲೂಡ್ ಮಾಡುವಂತಹ ಒಂದು ಕಾಲಮ್ ಕ್ರಿಯೇಟ್ ಮಾಡಿ, and he explained the metabolic character character of each and every group of this microorganism in a single column. So this is what we call Winogradsky column. I have not exactly explained in detail about this column. I am just giving you an idea here. So in later semesters in soil microbiology, you will be learning this in detail. The last scientist we have here is a Russian microbiologist, Metchnikoff. Metchnikoff was a Russian-born zoologist and a microbiologist. He was honored as the father of innate immunity because he was the first to discover a process of immunity in our body called phagocytosis. What is phagocytosis? See, this is a process where when a pathogen enters inside the human body, it will be first encountered by a group of cells called phagocytes. So one such phagocyte is macrophage, and these macrophages usually will engulf the pathogen, will destroy it then and there, and will alert other cells in our body like T cells and B cells. So these are the first line of defense that we have in our human body, and these macrophages was first discovered by Metchnikoff in 1882. And he received Nobel Prize along with Paul Ehrlich in 1908 for physiology or medicine for his discovery of this phenomenon called phagocytosis, especially in animals. And this discovery turned out to be the major defense mechanism in innate immunity. So apart from the work of immunity, Metchnikoff also worked on the concept of aging process in human beings, and he advocated the use of beneficial bacteria called lactic acid bacteria, which can be consumed for healthy and long life. In fact, ಈಗ ಏನು ನಾವು ಹೇಳ್ತೀವೋ probiotics ಅಂತ ಏನು ಕಮರ್ಷಿಯಲಿ ಮಾರ್ಕೆಟಲ್ಲಿ ಸಿಗುತ್ತೋ ಆ probiotics ಅನ್ನುವ ಕಾನ್ಸೆಪ್ಟ್ ಅನ್ನು ಫಸ್ಟ್ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ದೆ ಮೆಚ್ನಿಕಾಫ್ ಅವರು. He told that if you consume these beneficial bacteria on a regular basis, you will get a very healthy and a long life. And apart from that, Metchnikoff is also credited with coining the term gerontology, which is the study of aging and longevity process. So basically, whatever process that is happening in human beings, like immune system, how it works and how it results in aging process, how we can reverse the aging process by consuming some beneficial bacteria, ಇದು ಎಲ್ಲ ಟೈಪ್ ಆಫ್ ಹ್ಯೂಮನ್ ಸಿಸ್ಟಮ್ ಅಲ್ಲಿ ಆಗುವ ಎಲ್ಲ ಟೈಪ್ ಆಫ್ ಸ್ಟಡೀಸ್ ಅನ್ನ ಮಾಡಿದ್ದು none other than our Metchnikoff. So with this, I would like to end this video. Thank you.